All right, we're back. Thanks for sticking around. I'm sure you were so excited to get back into the cell cycle. So here we are. Now, Cole was talking to us about prophase. So while we were taking our break, I took a few minutes to make sure I understood it. So I'm going to go ahead and add that to my cell cycle on the whiteboard. And then again, our scientists will correct me if I'm wrong, but I'm pretty sure I got it. So I'm going to draw my other cell here.
Great start.
Okay,
Great
and then
start.
Cole took a lot of time to talk about how they were matching up. So I'm going to go ahead, have one matching there. Oh, whoa. Oh, got ahead of me. That's all right. And one here. And let's do one more right here. And Cole said that the nucleus, like the outside of the nucleus, the nuclear membrane, dissolves. So I'm going to leave it just like that. Is that good?
Honestly, Mackenzie, that is, I think, even better than mine. It looks really good. Oh, one thing I might add is those spin fibers on the outside.
I'll just get those started here, just like that. Perfect. Now, just to make sure we totally understand this whole matching up thing, I came up with a little game. I want to test the scientists, see who can correctly match all of these things correctly, like similar to what happens in our cells. So what we're going to do, listen carefully, scientists, this is going to be our game. Okay. So what I'm going to do, I have all of these objects here, each object, similar to chromosomes. There's two chromosomes that match up. So I'm going to go and I'm going to mix them all up. And I'm going to spread them out on the floor in front of us. And then one time, you're going to have 2 seconds. You have to start behind your desk. Then you're going to run up one at a time. Cole's going to go first. Okay,
You want me to go first?
Yep.
Okay,
Yep, you're going to go first.
I'll go first.
You're going to match them up as quick as you can. And then you're going to go back behind your desk. So if you finish before the 20 seconds, when you're done behind your desk, I'll mark down your time. If not, if you're still going, by the end of the 20 seconds, you need to be behind your desk, and then I'll count how many matched pairs you got. The person with the most matched pairs or the fastest time wins.
Okay.
Got it?
Yes.
Perfect. Okay. Any questions? Last chance.
I'm ready to rock.
I'm
ready
good to
rock.
go.
Oh, yep. So I'm going to sneak out in front here. We're going to mix these up real good.
Should we look away?
Yeah, maybe I'll close my eyes.
Yeah,
I don't want to, no spoilers here.
Yeah.
I have a joke for the meantime if you want, Cole.
Yeah, are we both closing our eyes?
Are we?
Are you closing your eyes?
I guess.
Tell your joke.
Okay. My joke is, so what do you say to your sister when she hits your foot?
I'm sorry that you hit your foot, sister.
You say, mitosis.
Oh, that is good. That's good.
Okay.
That's beautiful. Love that.
Wait a second.
Okay.
Vanessa, are you peeking?
No.
Oh,
I would never.
wouldn't be surprised.
Okay, well, I'm going to head back to my station.
Okay.
But I think that Vanessa might have peeked. So because of that, Vanessa, you have to go first now.
Oh, I'm so nervous.
Yeah.
Oh,
Okay.
That's how it's going to go.
nice.
All right. Well, you know what? Even though I wasn't prepared, I can do it.
Okay. Here we go.
'Kay.
I got my 20-second timer here. 10-second countdown, and then it's all you, Vanessa.
All right.
10, 9, 8, 7,
This is
6,
intense.
5, 4,
Whoa.
go.
Okay.
Okay, here we go.
So she's got to try to find the matching objects. That's how we're working this?
Yep.
Okay,
That's
okay.
right. She's got 10 seconds left.
Oh my goodness, that
I'll
went
give
so
her a 5
fast.
second countdown to make sure she's behind her desk.
This is hard.
5,
This is hard.
4,
Oh my goodness.
3, 2, get behind your desk. All right.
Whoa.
The timer even said well done.
Whoa.
Oh,
So,
thanks, Timer.
I'm just going to take a second here to count them. We got
She looks like she did really well. I have to say.
1,
Thanks, Cole.
2, 3, 4, 5. I don't think these ones are quite matched up, so I think Vanessa has 5.
Ooh.
Now I'm going to go ahead, close your eyes
Yep, of
for
course,
sure. If
of
you don't
course.
close your eyes, Vanessa, you're going to get a penalty for
Uh-oh,
sure.
uh-oh.
Okay, last warning. So I'm mixing them up again. Here we go.
I got a lot resting on this.
Yes.
This is my
This is-
pride.
this is a
You
big
on the
really
one.
line
do.
here.
Can use your magic.
All right. Here we go.
Okay. I promise I'm not looking.
Okay. Sneak back here.
Yeah.
Got it. We're ready. Are you ready, Cole?
I'm so ready. I've never been more ready.
Okay, we got another 10-second timer going.
Okay.
Are you ready?
Okay.
Okay. 10-second countdown. 8, 7,
Oh boy.
6, 5, 4. go.
Okay, here we go.
Here we go.
Okay, so
Got
these
this
are
cool.
the same, and I think this kind of looks the same as this, and then this is gonna go here. Oh boy, oh geez. Okay, lots of pressure, lots of pressure.
You got
And
it,
maybe
you're
we'll
going
put these
10
together,
seconds.
and then this one. Oh no, where did it all go? Where's the other wheel? Oh,
Oh,
this is a disaster.
5,
okay.
4.
I'm just gonna get it back. I just don't wanna, I don't wanna lose.
All right, well,
Okay.
I'm going to go and
Beat that,
inspect.
Vanessa.
You might have.
Okay, so we have 1, 2, 3, 4, 5, 6. Okay. Something happened here. So we have
That's a pair, isn't it?
a pink one and an orange one. Those don't match, Cole. What happened? Can you explain
Oh,
yourself?
Sorry. Okay, Mackenzie, I know what happened here. I did that on purpose. All right? I was trying to explain that when we talk about mitosis, we talked about how these chromosomes pair up. Now, we did it really well here where I had my red ones and my purple ones and my orange ones all mixing up together, but that doesn't always work out perfectly. Sometimes our cells, just like us in real life, make a mistake, and maybe the right chromosome or a piece of the right chromosome doesn't match up with its partner. Now, when this happens, we call this a mutation, and there's lots of different mutations that can happen inside of our cells. For the most part, mutations are happening all the time in our cells, but they don't have any kind of consequences, whether that's positive or negative. They don't really do anything because a lot of our DNA actually doesn't even really code for anything. It's just kind of there. So if a mutation happens in those parts of our DNA, we're okay, everything's fine. But sometimes a mutation can happen where maybe we are, when we're conceived, there is more or less than that number of chromosomes that we want, which is 46. So we talked about how most somatic cells, which are our body cells, have 46 chromosomes. If you have more or less chromosomes, you might be born with some sort of genetic deficiency, okay? Now, if you're born with more chromosomes, like instead of 46, maybe 47, you might experience something like Down syndrome, for example. That's caused by an extra copy of a certain chromosome. So these mutations can happen. Sometimes our cells aren't perfect, and they do sometimes make mistakes.
Perfect. So before we get on to the next phase, I just want to remind you that if you have any questions, you can feel free to text us at our number 306-1013. You can also reach us on social at @IsYouth and use the hashtag CouchPotatoLab, and we'll be sure to answer your questions there. Now, Cole, I'm really glad you were just talking about those mutations and stuff because we actually got our first question from a viewer. And Megan asked, can you have more or less than 46 chromosomes? And I think the answer to that question kind of relates to what you were just talking about. Do you have anything else you want to add about that?
Yeah, so like we were saying, you can be born with more chromosomes. There's actually not a case where you can be born with less chromosomes than 46, but you can be born in 3 different cases with an extra chromosome. So instead of 46, you would have 47. And there's 3 different cases where that would happen. And each time an individual born with that extra chromosome is going to experience some sort of genetic deficiency of a wide variety of things, okay? But for the most part, those people live really, really happy and full lives, and everything is a-okay. But that can happen, and when that happens in humans, what happens is instead of having 2 copies of each chromosome, one from mom, one from dad, you actually have 3 copies of a chromosome. So that might be chromosome number 18 or chromosome number 21, and you have an extra copy. Instead of the normal 2, you have 3. So we call it a trisomy, tri for 3. So yes, that can definitely happen. It happens all the time. But like I was saying before, most of the time, mutations that happen inside of our cells don't have any consequences. They're neutral mutations, which means that they don't really do anything bad and they don't really do anything good.
Great.
Yeah.
Thanks for adding that, Cole. Now, Vanessa, I think I'm ready for the next phase. Can you continue with the next phase, please?
I absolutely can. So our next phase is called Metaphase. It is when the because we've been talking about the spindle fibers before. Those are these little things right here, and they're actually attached to these things called centrioles. So those are kind of the things that help pull everything apart when we get there. So the chromosomes like to line up on the spindle fibers right in the middle in a nice line, like so. So they just get like this and then get ready to pull apart. So I'm going to use a magnet to show this a little bit. So we have our magnet right here, and what would be really good on the spindle fibers, it kind of works like the magnet with the chromosomes where it gets pulled together, so even just like that. It just snaps right in. And then we'll move on to our next phase.
It's like a 2-for-1 deal.
Exactly. It's anaphase. Mackenzie, if you want to draw the metaphase up there, you totally could while I just get anaphase prepped.
Sure, sounds good. So I'll go ahead, and I remember there was still the cell. Now there was those, she said that they lined up in the center, so I'm going to leave them right in the center of this cell. And then I got my yellow, and they're still in their pairs, is that right?
Yes,
Yeah,
that's
okay.
perfect.
Perfect. And then Vanessa talked about that spindle, so I'm going to connect that spindle a little more. I'll leave it in the same color. And that spindle kind of went all the way out on each side. So I'll add that. Perfect. I think I got the gist of it. Is this looking good?
That looks great, Mackenzie.
Fantastic. Yeah.
All right. I'm ready for the next phase that kind of intertwines with this one.
Excellent. So
Great.
once we're done with metaphase and they're all lined up, it means that they're ready to get pulled apart. So it's really important for our chromosomes to get pulled apart properly so that our two cells can have the right amount of chromosomes. So that will happen kind of like when we had our magnet coming together, if you flip your magnet over, suddenly they're going to repel each other. So you're going to go like this and they just won't come together. If you've ever had a magnet before and you know what it feels like when you're pulling them together, you know that it's resisting and they don't want to be together anymore. They want to go apart. So that's exactly what happens. In this drawing here, we don't want that one to happen, but yeah, we have our chromosomes and they line up like so, and they're getting pulled apart. And that's just, this phase is really, really short, so it wouldn't stay like this very long. It just gets pulled apart all of a sudden, and the spindle fibers take it back to separate sides of the cell.
All right. So I'm going to put that on the whiteboard here as well. So I'm going to draw the cell. And then Vanessa said they get pulled apart. So I'm going to put half of it here, half there. I'll draw the one on the bottom as well. And then same thing with this chromosome. It's getting pulled apart. And it's still attached to the spindle, is that right?
Absolutely, yes. It needs a spindle to help pull it back.
Okay, so I'll go like this and I'll attach those spindles there. Just like that. How does that look?
That looks really great. Excellent.
Perfect. All right, so we know interphase, prophase, metaphase, anaphase, and you can see that it's working in a cycle for the cell cycle. Now, Cole, can you finish this off? Let's go to the next phase.
I can finish it off. So the next two phases are going to complete our cell cycle, and you should be seeing that at the end of it, remember we started with one cell at the beginning of mitosis. At the end of this, we're going to have two cells. Now, what's really, really cool that we haven't really touched on is these two new cells are also going to be identical to that first cell. All right, so why is this important? Well, let's think about our skin cells or our hair cells or our muscle cells. If we get a cut, let's say, and we want to heal, well, mitosis is going to let us heal. And we want to make sure that when we build new skin cells that go over that cut, we're actually making skin cells. We want them to be the same as the skin that's around it, right? So we don't want to randomly start making hair cells or nerve cells or muscle cells where our skin should be. So it's important that at the end of mitosis, those two new cells that we make are genetically identical to the one that we started with. So the last two phases, the first one that we're going to get through is called telophase. Now, like Vanessa was saying, in anaphase, those chromosomes are getting pulled apart. So we paired them up at the beginning so that we had all of our pairs, all of our ducks in a row, per se. And when we get to anaphase, those pairs are getting pulled apart. So when we get back to our telophase here, now I got a chromosome kind of falling off here. But when we get to telophase, what you should see is a couple different things. You'll notice that I drew two new dotted circles on the inside, and that is our nucleus. If we remember back to the beginning of mitosis, we had our nucleus that was keeping our chromosomes safe and cozy on the inside. We're starting to reform the nucleus now, but not just one nucleus. You should see two new nuclei, I should say, forming. And inside those nuclei are our separated chromosomes. Now, I made sure that when I did this, I put one purple in each one orange in each one, and one red in each one. Because again, those were paired up before, and in anaphase, we actually pulled them apart. So in telophase, what's happening is those chromosomes are going into two new nuclei. the cell is also starting to split. That's kind of what we're showing with this lovely diagram here, is the indentations in the middle are showing that the cell is going to very, very soon, possibly in the next phase, split apart, all right? So that stuff on the outside of the nucleus and in between the cell membrane and the nucleus is called cytoplasm. And what's happening is, as we prepare for that final kind of separation, the cytoplasm is starting to split right before our next phase.
All right, so I'm going to go ahead and draw that next phase here on the whiteboard. So we're going to draw the cell, and Cole talked about that little indentation. So I got that there. Now he also talked about the two nuclear membranes forming. So I'm going to go ahead, draw 2 dotted circles here. And then I'm going to make sure I put the same color. So I have 2 oranges and 1 yellow. So I'll put 1 yellow on this side, 1 in this side, and then 2 oranges in each of these as well. Perfect. Now I think that looks pretty good. And Cole mentioned something about cytoplasm, and that is actually part of the cell. If you want to learn more about the cell, we did an episode on that. That's back in episode 11, which actually brings me to one of the questions we got from one of our viewers. They said, "Hi there, I'm Megan. Can the Eyes team sing the cell song?" Now, I would love to sing that song. I had so much fun doing that rap, but unfortunately, my other trio, the 2 people to make up the trio, aren't here today, and I would hate to steal the fame from them. So if you want to see that cell rap, just head back to episode 11 and check that out. Great. Now let's finish it off, Cole. Let's finish off with the very last phase.
Let's do it. Let's do it.
Great.
So the very last phase is called cytokinesis. Now, if you remember back to that word cytoplasm, the prefix cyto, C-Y-T-O, that kind of means cell, and kinesis means movement. So what we're actually seeing in cytokinesis is cell movement. The cells are actually going to separate. So if we imagine for a second that, and I know Mackenzie was using some Play-Doh before, we have even more Play-Doh that I'm going to use. If we imagine for a second that this ball of Play-Doh is our cell that has just finished telophase, at the end of telophase, it's going to be kind of squeezed out, a little bit elongated, kind of like this, sort of looking like a peanut, if I can try to do that. A little bit. So our cell is elongated, and in cytokinesis, what's going to end up happening is we're going to get, it's going to keep getting indented and indented in the middle until finally, bam, we split. And at the end, we have our 2 new cells, okay? Now remember, Mackenzie talked about before exponential growth. This is going to continue happening, and you can see why the number of cells in our body is going to get so high, is because when this splits and this splits, you're going to have 2 and 2, and that's 4, and then 8. And Mackenzie talked about how that number goes up really, really quickly. So in cytokinesis, if we look at our diagram, the finished product of our whole mitosis adventure here is going to look like this. We have 2 new cells that are genetically identical to the cell that they came from. They have all the correct chromosomes and genetic information. Both of them are going to have, we're only showing 3 chromosomes here, just so that it's easier to see, but you can imagine that there's actually 46 of these in each. And our nuclear membrane, our nucleus, has fully reformed. So that smaller circle on the inside of the cell has fully reformed, keeping all of the DNA and the chromosomes safe on the inside. So after all of that, interphase, prophase, metaphase, anaphase, telophase, cytokinesis leads us to 2 new cells at the end of mitosis. We did it.
Wow, all right, what a ride. Now I'm going to just put that last phase here on the board so we have our entire cycle. So we have 1, and you can see I'm going to draw a completely separate cell because at the end of the cycle we have 2 brand new cells, and I'm going to go ahead and have them here. Put that DNA in them again because it's a complete cell. And although they're not perfect because my artistic abilities aren't perfect, you can see that they are genetically identical. And throughout this all, I have been drawing arrows. And that's because this is a cycle. So as Cole was saying before, this cell now is just going to go to here, and it's going to start the interphase, prophase, metaphase, anaphase, telophase, back to cytokinesis. Same with this one. So all the cells just keep dividing, and that is how we get our exponential growth. Now, before we go any further, I just want to remind you that you can ask all of your questions by texting us in at 306 570-1013. And you can also reach us on social at @IsYouth using that hashtag CouchPotatoLab. So you can find us on Twitter, Facebook, Instagram, all that great stuff. And I have another question. We got one from our viewers. It says, where did you get all that Play-Doh? And that's coming to us from the Hayes brothers. Now, I'll actually answer that. It's kind of a long story, but last night I was trying to cook supper and I was mixing some things together. It wasn't going so well, so I actually made it at my house. I transformed my supper and it turned into Play-Doh.
Wow.
Wow.
Mackenzie, well, thank you. We needed it today, so I'm glad that happened.
Me too. I am very, very glad. Now, we've been talking a lot about how mitosis happens in humans. And I was thinking, how did I start as a tiny little baby, and then a toddler, then a teenager, and now I'm here all grown up on the Couch Potato Lab? And I started thinking, talking to my friends, I started seeing their baby pictures, and I was thinking, how did they become who they are? Now, Cole, that must be related to mitosis.
It sure is, Mackenzie. That's right. So, as I'm sure you all know, we didn't just start at the size that we are right now, okay? We started very, very small. You might think we started as a baby, but even before that, at some point in time, every one of us was one single cell. And then through the process of mitosis, that cell split into 2 and 4, like we talked about earlier. And that number keeps growing and growing and growing. And as we develop and as we get older, our body parts start to specialize and grow. And we become how we look like today. So that's pretty cool. It's mitosis that is actually responsible for us growing. So when we look back at old pictures of us, for instance, at our baby pictures or pictures when we were younger, we've experienced a lot of mitosis in our bodies since that time.
Perfect. Now I've been thinking and I've come up with our science showdown. So here we go. It's time for our science showdown. Great. Now, today we have an extra special science showdown. So what I'm going to do, it's going to be a competition between the lovely scientist Cole and the lovely scientist Vanessa. And what we're going to do is I compiled all of these baby pictures from the EYES team. So all of the people who you've seen on the Couch Potato Lab, I got all of their baby pictures. And we're going to do our best to try to guess who is in the picture. And we're going to have to use clues of who they are today, and we're going to have to try to think back to before all of that mitosis happened and guess who it is. Are you ready?
Yes.
I think I'm ready. I'm ready.
All right. Okay, so we're going to start here. We're going to show the picture here on the screen. You're each going to get one guess, and we're going to try to get the winner. All right, picture number 1.
Okay.
Who is it? You have 3 seconds to look at the picture.
Okay.
3,
I'm looking.
2,
I'm studying.
1.
Fiona.
All right. I heard it. What did you say?
I said Fiona. I thought maybe it looks like a blonde baby a little bit darker hair, like a darker blonde. That's what I'm thinking.
Okay, Cole, what's your guess?
Listen, there's only one name that comes to mind when I can't think of anything else, and that's Tom. So I'm going to guess Tom.
All right. Well, turns out that picture was indeed Fiona. So that is 1 point for Vanessa. We got a point for Vanessa.
Perfect.
All right. Picture number 2. Here we go.
Okay.
All right. And we have 3, 2, 1. All right. And cool. Your guess first this time.
Okay. Well, this one actually, I have a very strong feeling about this one.
Okay.
I'm thinking, and it's only just because I feel like I've seen this person recently.
Okay.
I think that is Vanessa.
Whoa, Vanessa. Cole guessed Vanessa.
Yep.
Well, Vanessa, who do you think it is?
I think that one's me. I remember my fairy wings, and then that's also my mom in the picture. So.
Okay. Well, turns out you're both correct. It is Vanessa.
Yay.
So we have scores. That's a point for Vanessa, so 2, and it's also a point for Cole. So we are at 2-1.
Okay.
All right.
I can come back. I can make it back from this, I think.
We'll see about that.
I'll keep those on my fingers so we don't forget. Now let's head to our third picture. All right, here we go. And 3, 2, 1.
Okay.
All right.
I got this one.
Vanessa, we're going to take your answer first. Who do you think it is?
I'm a little bit more lost on this one. I'm going to go with Cole's typical answer of Tom.
Okay, we got Tom as a guess. Now, Cole, what's your guess on this one?
See, I know why you would say Tom, and that's because he is always wearing that exact same onesie to this day, but it's actually not Tom. I believe that is Sabrina.
All right, and the answer's in. It is Sabrina.
Yes.
That is a point for
Yes.
Cole. All right. And we'll move down to the fourth image.
So we're all tied up, 2-2.
We're all tied up. We
Okay.
got two
Oh,
for
perfect.
2. Look at that.
All right.
Okay. Here's our fourth image. All right. We'll get that on the screen here. And you got 3, 2, 1. We'll
Huh.
take Cole, your guess first.
Okay, this one looks tough, but I'm trying to think of somebody who would like baseball. I saw a baseball bat in the picture. I'm the only person that I can think of that loves baseball is Mackenzie. So I'm going to go with Mackenzie.
It's true. I do love baseball.
You do
That
love
is
baseball.
a very great guess. All right, Vanessa, do you have a guess for this one?
I think that one is Cole.
Cole?
What?
You think Cole doesn't know that it's Cole?
I don't know. I guess we'll find out.
Well, let me look. Ooh, the answer is Cole. That
Oh,
is
no.
a point for Vanessa, and I think Cole needs to pay a little
Oh,
bit more
man.
attention.
That's gone from my memory. I don't remember that at all.
I don't know what
Crap.
to tell you.
Okay.
Maybe you were a baby.
All right, let's move on. We have 3 points for Vanessa, 2 points for Cole, and we're
Mhm.
moving on to our fifth picture. And we're going to look at that. 3, 2, 1. All right, we're going to start with Cole. What's your guess?
Okay. I definitely know this because not only do I know that Mackenzie likes baseball, but I also know that Mackenzie likes dogs. So I think that one actually is Mackenzie.
Okay, good.
That's my guess.
Pretty fair guess. Vanessa, what do you think?
I'm thinking because of the blonde hair, I think it is you, Mackenzie.
2 guesses for Mackenzie. Well, turns out that it is me. It is
Yes.
Mackenzie. So we have a point for Cole and a point for Vanessa. So our score is 4 for Vanessa, 3 for Cole. Oh yeah, that was a tricky round. All right, now we're going to move on to picture number 6. All right, let's see what we can do here. We'll put that on the screen in 3, 2, 1, and we'll take Vanessa's guess first this time.
I'd have to say it is Covey. I saw a little hint.
Covey, you saw a hint. Interesting.
Mhm.
Cole, what do you think?
This one is tough. I was studying it very closely, but listen, there's only one guess that I can possibly go with, and that's Tom. Is it Tom? There's got to be one of these that has to be Tom, right? Right?
Well, turns out the answer is Covey. If you looked very closely, it actually said his name on the pant leg. So,
Unbelievable.
Cole, you got to step it up here. That's another point
Dang
for
it.
Vanessa, and we have 5-3. All right, and we're going to move on. Here we go. This is our final picture.
Oh,
And this is a bonus round. It's for 2 points.
whoa, a bonus
Okay.
round.
You could
So
catch
we're
up,
at
Cole.
5-3. We could catch up, but I don't know. Can you do it?
I don't know about that.
All right, our 3 seconds are passed. Let's take Cole. Your guess first.
Oh, geez. Okay. This is for all the marbles, isn't it?
Yeah, we
It is.
5-3.
keep in mind we haven't had Tom yet.
That's very true.
You're right. We haven't had Tom yet.
No.
It has to be Tom. I'm gonna guess Tom. It's got to be Tom.
Okay, you better hope it's Tom because you're really relying on those 2 points to tie it up. Vanessa, who do you think it is?
I'm going to have to diverge from the Tom guess. I think it's Katie.
All right.
Yeah, right.
I have to say it is Katie.
Yes!
So Cole,
What?
you're stuck at 3 points, and Vanessa, she has more than my hand, so she has 7 points.
More than
and,
your hand.
And our winner, here it is. Let's see. All right. I just gave you a huge hint there. It is Vanessa. She wins our science showdown today.
Congratulations.
So Vanessa, you started as that little girl,
Yep.
and through mitosis, you have divided your cells and grown up to be the beautiful lady that you are
Oh,
today.
thank you. I wish I still had my fairy wings.
We'll get you some of those. Don't you worry.
Perfect.
Now, that was great. I loved that so much. But even more than I love Science Showdown, I love Ask a Scientist. And it is time for that segment right now. All right, so just looking at what the headquarters is sending me here, we have a couple really great questions. And the first one comes from the Hayes brothers. And this one's for you, Vanessa. They want to know if you can use bleach instead of alcohol when you're doing a DNA extraction. Vanessa, what do you think?
Great question, Hayes brothers. You actually can't, unfortunately. We really need this isopropyl alcohol because it is used to pull and precipitate the DNA out of the mixture of strawberries and dish soap and everything. It's the polarity of the alcohol. So different charges in the alcohol versus the dish soap and the strawberries and the DNA. It all comes together, and you unfortunately need isopropyl alcohol.
All right, there you have it. You definitely need to use the isopropyl alcohol. Now, Cole, this question is for you because you're the chromosome expert.
I am.
So we have a question here from Theo, and Theo wants to know, do all animals have the same number of chromosomes as us as humans?
That is a fantastic question, Theo, my friend. The answer is actually no, not all animals have the exact same amount of chromosomes. So, like we said before, our somatic cells, which is our body cells, they have 46 chromosomes each under normal conditions. But there are lots of different other chromosome counts that you can find in animals. And I'm just going to take a look. I have a note here from headquarters, a couple different things. So some interesting stuff. A fruit fly, for example, has 4 pairs of chromosomes, all right? Only 4, where we have 46. A rice plant has 12, and a dog has 39. So there's lots of different chromosome counts that we could get depending on the species. And what's interesting, what you should keep in mind is the number of chromosomes. The higher the number, that doesn't mean the better or the more complex that organism is. The number of chromosomes doesn't really have anything to do with that. It's just the different information that's coded on there to tell a cell to become one day a human or to become a plant or a dog or whatever. So yes, the answer is lots of different animals, plants have different numbers of chromosomes.
Yeah, now a really cool thing about chromosomes too is we as humans, we're not the only species that has 23. For example, there's actually a small species of deer that actually also has 23. So it's not just reserved, it has different information in that DNA. So that's what makes it really different is those instructions in that DNA would make it the 23 pairs either the human or that small species of deer. So that's really great as well. Now we have another question here. And it's also coming from Theo. And Vanessa, Theo wants to know, do all cells undergo mitosis?
No, not all cells undergo mitosis. Some cells go through a process called meiosis, which sounds very similar, but it's pretty different. It happens when we're reproducing, so the cells will come together and they actually exchange parts of their chromosomes. So it's not identical because it's coming from 2 different organisms. So it exchanges parts of the chromosomes and it makes new cells. So all the cells are not identical. It actually doesn't make 2, it makes 4. Cells, so it's pretty cool. And some cells that do go through mitosis, they actually just stop at some point. That's the G0 phase. It's like a side phase. So some cells will just never go through mitosis again. So yeah, no, not all cells go through mitosis, even though it's really cool.
Great answer, Vanessa. Now maybe we'll have to cover that in another episode. We're getting so many great questions from all of you viewers. We're so thankful that you're tuning in and giving us these great questions. We have another really awesome question here. It says, why do you draw your DNA like squiggly lines instead of straight lines?
That's a great question. Now, part of it, there's a few different reasons. I think one part of it is if you were to actually look at a microscopic image of a chromosome, something that we didn't mention is most of the time when our cells are in interphase, they spend a lot of time in interphase just chilling out, maxing, relaxing, all cool. But what happens is the chromosomes at that point are actually really, really uncondensed, and they're almost like invisible. But when they go through mitosis, they condense and they clump up. And if you were to take a look at a microscopic image of a chromosome, it kind of looks all squiggly like that. Another reason is if we were to zoom in even further on those chromosomes and look at the individual DNA molecules. If you've ever seen a diagram of DNA, it's a really, really common model or diagram that you might see in a science classroom or something like that. DNA is in the shape of a double helix. Now, I don't know, Mackenzie, do you think you could draw a double helix?
Put me on the spot like that?
On the board?
Let's see.
Just so that we can see? Because the double helix sort of is a spiraled shape.
Yeah. So let's try. I have this.
That's a good start.
Okay. Should I do it in the same color or different color?
Maybe do the 2 strands in the same color, and then the ladder you can do in a different color.
Good idea.
Maybe
Yeah.
this. Okay. And then the ladder. Okay.
So what Mackenzie's drawing there is those are our 2 strands of DNA, and those little green bits in between are what are called DNA base pairs, all right? And we use different letters to represent those, A, G, T, or C. And those base pairs match up, and when our cells are learning how to make things, when they're making different things for our body, there are special machines inside of our cells, special organelles that read that DNA just like you would read a book, and they tell our cells what to do. So there's a lot of different reasons why we draw it kind of squiggly. There's the DNA double helix. There's also just, that's kind of how chromosomes look.
Perfect. Now, Rhythm, one of our viewers, wants to know, when our cells get pulled apart, do we feel it? Vanessa, can you answer this for us?
Thankfully, it doesn't hurt. We can't feel it. Doesn't feel like anything because it's so tiny. You need a microscope to even see one of your cells. So seeing the even tiny parts of chromosomes, you need a microscope, and you need pretty high power on the microscope to be able to even see it. So you can't feel it at all. No.
Well, that's a relief. I am glad that I don't have to worry about feeling that. Now we have one more question. Now, Eli has been wondering this for days, and I am so glad that we finally have enough time to get to this question because it is a great one. Now, Cole, why does your voice sound different when you breathe in helium? That is the burning question, and Cole's going to give us the answer right now.
So what a great question. Sort of off our train of mitosis, but that's okay. We love all types of questions here at the Couch Potato Lab. So the question is, why does helium make your voice sound really, really high-pitched when you speak? So if you've ever, and we don't necessarily recommend doing this at home, but if you have seen somebody else, maybe on a video or something, inhale some helium and then speak, you'll notice that their voice is a lot higher pitched than it normally is. Now, the reason that is because helium is a gas, and helium is a gas just like the air that we breathe around us is a gas. But the difference between the air that we breathe and helium is much less dense, okay? So a way to think about density is just the amount of stuff packed into space. So when we talk about helium being less dense, all the molecules are really, really spread apart, and it's almost like it weighs less than air. So when we breathe in helium and it fills up our lungs, and then we speak that air back out through our vocal cords, because there's a difference in density between the helium and what the density of normal air usually is, you're going to get the sound waves vibrating a lot differently in our vocal cords. So with helium, because it's less dense, those sound waves vibrate a little bit differently and they come out a lot higher pitched. Now what's interesting, and you might not have known this, is we can actually do the opposite way too. If we were to take a gas and inhale it that's a lot more dense than air, so it has a lot of molecules packed into a tight space, and we were to inhale that, our voice would go a lot lower instead of higher. So for example, there's a gas called sulfur hexafluoride, which is 1 sulfur atom and 6 fluoride atoms. And when we take that, we inhale that, it's going to do the opposite of what helium does. It's going to make our voice a lot lower. And again, that's just because the different density of the gas that is being pushed through our throat, through our vocal cords, causes those vibrations to change, all right? So that is the answer to the burning question that we've been waiting for so many weeks now. That is why helium makes your voice higher.
Thanks for that, Cole. Now that ends our Ask a Scientist segment, but there are definitely a few viewers that have been waiting for this announcement. Now this week we've been having a giveaway, and we've been wondering, what is the in-studio elephant's name? Now we had 2 of our friends, they had the right answer, they sent it in to us, and the right answer was Effie. Effie the elephant is our in-studio elephant. Now our winner is, drum roll quick, it is Eli. Now Eli, you're going to email us, get your parents or guardians to contact us, and we'll be sending you this potato clock and some ice swag. I'm sure you're going to love it. Now, next week, we'll also be doing a giveaway, so tune in to our episodes next week, and we'll be having a lot more fun things to give away. Now, before we go, I just want to send a huge thank you to all of our supporters and to you at home for joining us each and every day. We love interacting with you. We love bringing the science to you. And we are so grateful that you're asking all of these amazing questions. That's it for us all today. So from us to you here at the Couch Potato Lab, bye and see you later.